Good evening everybody. This would be another small session, second one on pharmacodynamics. Last time we talked about what is pharmacodynamics, that is how the drugs they act, details of mechanism of action of drugs, and I told you there is a difference between action and effect. We have to remember that action is when a drug binds to the target organ, then what happens in the target, that is action. And effect is ultimately what are the changes in the biological system overall, that is effect. So effect is a bigger thing. Action is immediately what happens as a consequence of binding of the drug to its target. Last time we talked about some mechanisms which are non-receptor mechanisms, not mediated through receptors. There also I told you that we should not take them lightly. They are rather in majority. Most of the drugs they act without acting on receptors. So what is receptor? What is the region? Because in certain books you will find it is written more than 90% of the drugs they act on through receptors. It is because the definition of receptor. A receptor we can say is a macromolecule that invariably is a protein. So a receptor is a macromolecule that binds to the ligand and starts action. All these criteria, if they are fulfilled, then only we should call it a receptor. I give you example of plasma proteins. They also bind to the ligand, they also bind to the drugs, but we do not call them receptors because there is no action is started. So sometimes we call them inert receptors, but sometimes we call them acceptors, but they are not receptors. Other examples I gave you last time, like say in enzymes. Enzyme they bind to the ligand, they bind to the substrate and they produce the effect, but they are not regulatory proteins. They bind and because of that the action takes place, that is true. But if it comes strictly to the receptors, then all of these functions should be fulfilled. In enzymes I told you that if they are situated outside the cell, the action will take place there. If they are situated inside the cell, the action will take place there. So these are the functions of receptors. Yeah, there are receptors which are intracellular receptors, cytoplasmic receptors, but there also the effector domain, it has some regulatory functions. So a receptor should transfer the signal. Actually we can consider that a drug is a signal, is giving a signal. This signal needs to be identified and converted into some effect. That is the function of a receptor. So a receptor should transfer the signal across the plasma membrane because most of the receptors are situated on the cell surface. A receptor should do transduction. Transduction is conversion of this signal into action. That is transduction. Conversion of this signal into effect, that is transduction. Should be able to amplify. Suppose the signal is less, there may be more effect. Should be able to integrate. There may be signals from various sides and all of them they converge into one. So all these are functions of receptors. This is another question that is asked, what are the functions of receptors? So in nutshell we can enumerate them. Receptors are those which transfer the signal across the membrane, which causes transduction, which can amplify, can integrate the signals. So these are the functions of the receptors. These criteria are not fulfilled by enzymes. Enzymes can be a part of the receptor system. Actually receptor is a system. So enzymes can be a part of receptor system, but alone enzymes cannot be called, that is I give you example of say tyrosine kinase. At many places you will find it is written non-receptor tyrosine kinase, means tyrosine kinase is available as enzyme also, but it is a part of receptors also. Kinase receptors, tyrosine kinase receptors, insulin receptors, they are, because there is a binding site at particular place. So we have to remember this. When it comes to receptors, we have talked about, so in receptors, to fulfill those functions, there are various possibilities. One is that the ligand is something that can bind to the receptor, that can be identified by a receptor, and receptor can bind in, then we call it a ligand. So a ligand is bound to the receptor, and then what happens as a consequence of that binding? So one possibility is that the ligand will bind to the receptor and there is 100%, I would call it intrinsic activity. That is another quality of a receptor. Intrinsic activity, I talked about action. So that is intrinsic activity that will happen if the full agonist bind to the receptor. So if the ligand binds and there is full intrinsic activity. Another possibility is it binds but there is no activity, no intrinsic activity. Third possibility can be that the ligand has bound but there is activity, not much. And fourth, the ligand has bound but activity also has taken place, but it is opposite. All these possibilities are there in case of receptors. And accordingly, the ligand is classified as full agonist, partial agonist, antagonist, and inverse agonist. Basically, we have to remember that for every receptor that is present in our body, there is some endogenous ligand. Nothing in our body is without any purpose. Though we know about certain receptors for which we don't know what is the endogenous substrate for that, what substrate binds to them. But that is our inability. We call them orphan receptors. It is our limitation. We don't know which substrate binds to them. It does not mean that they are without any purpose. So inside our body, every receptor has an endogenous substrate, and normally this is the agonist for this receptor. This is actually one of the definitions, that response to the endogenous substrate of the receptor is agonistic activity. And this is how we identify what is the normal activity of the receptor. The normal activity of the receptor is what happens as a consequence of endogenous substrate binding to this receptor. That is how we come to know. And remember, there are so many types of receptors that we know and we classify them. So how do we classify them? There are various types of classification, pharmacological classification. Now actually we have found out the genes also. We know exactly what happens. So there are chemical classifications, but we have to remember that all these classifications are considering that what is happening physiologically inside our body. If I have alpha receptors, I have adrenaline that will bind to it and produce effect. So this is physiological activity of the receptor that we take as consequence of agonist binding. So this is how we come to know. So when the receptor has full affinity, this is the term we call affinity. Affinity means desire to bind to its ligand. That is affinity. And another is intrinsic activity, means action as a result of this binding. So these two things, based on these two things, we divide them. So if there is 100% affinity, 100% intrinsic activity, we call it full agonist. If there is affinity, 100% affinity, but there is no intrinsic activity, then it is antagonist. Now I am contradicting myself. In case of plasma proteins, I said we don't call them receptors. Okay, so that I should call it antagonist. But remember, antagonist, ANT I have added in front of agonist. That means antagonist can be there only if we know about some agonist. If we don't know about any agonist of any receptors, I cannot say this is the antagonist of this receptor. So this is relative. I have a receptor, I know the full agonist for this receptor, and now I add something, some chemical binds to it, but there is no activity, I will call it antagonist. It is not happening in case of plasma proteins. I don't know about any ligand that binds to plasma proteins and produces an effect. So that means they are inert. They do not respond to any molecule. Here it is a receptor, and it is a receptor has been proved by full agonist. And then if some ligand binds and does not produce any effect, then I will call it antagonist. And this is, there is full affinity, but intrinsic activity is not full. The effect is not full. And so what happens in older books, I remember Satoskar, there was no KD Tripathi at that time, what was written, partial agonist acts as an antagonist in presence of full agonist. And I could never understand what does this sentence mean. So it's basically simple. If I have a receptor that is bound, I have a bound agonist, full activity is going on, and then I add some partial agonist. So this partial agonist will replace some amount of agonist, because this also has affinity, 100% affinity, and because of that this partial agonist is having lesser activity. So overall activity will decrease, because half of the activity is because of full agonist, half of the activity is because of partial agonist. So effect will decrease. So what should I say? When I added partial agonist, the activity decreased, and activity decreases because of antagonist. And so we say that in presence of full agonist, partial agonist behaves as an antagonist, but in absence of full agonist, partial agonist is an agonist, because some activity will take place because of partial agonist also. Antagonist, no activity at all. And never get confused. We always confuse that the antagonist will have this effect, the antagonist will have this action. Antagonist does not have any action, cannot start any action. It does not have intrinsic activity. So what we see as a consequence of giving, when we give propranolol, we say heart rate decreases. So it's not that propranolol is doing something so heart rate is decreasing. It is because the action of agonist in our body, that is noradrenaline, it is no more there, because propranolol has blocked the receptors. So propranolol is not doing anything. It is just not allowing the norepinephrine to work. That is, so antagonist does not have any intrinsic activity. Inverse agonists are basically just theoretical purpose, and we have DMC and beta carbolins. So it happens in case of those receptors which are having normal physiological action without binding to any agonist. They have intrinsic activity of the receptor. In that case, if some ligand comes and binds and this activity of the receptor decreases, this is possible in case of certain channels in the brain. So they have some receptors are there which are responsible for, like for maintaining my mood. When beta carbolins bind, then rather than inhibiting those receptors, when the activity goes in opposite direction, suppose I give benzodiazepines. So what happens? Sedation. And I give some other drug that increases the activity, causes convulsions. So that would be inverse agonist. There is effect. It is not that there is no effect. There is effect, but the effect is opposite. to benzodiazepines. Conversions are taking place. But as I told you, we do not have any medicine. We do not have any drug that we can give example of. Beta carbolines are only for practical purposes, DMCM, dimethyl. So inverse agonist, remember, there is intrinsic activity but in the opposite direction. Then we come to receptors. How do we classify them? I told you pharmacological classification, but most important classification is based on the transduction mechanism. One is based on structure also. Transduction mechanism is, as I told you, how this signal is converted into the action. That is transduction mechanism. So this is one classification. Structure wise also, when the receptors, they have different transduction mechanism, they have different structure also. So based on the function of the receptors, the very common classification is we classify receptors based on transduction mechanism or effector mechanism. I can call it effector mechanism also. And that way we classify receptor into 4 or 5 types. One is G protein coupled receptors. They are in abundance, very common. We have ligand gated ion channels. We have kinase receptors or kainate receptors, enzyme linked receptors, and we have cytoplasmic receptors. Actually, kinase also, they are of 2-3 types. They are enzyme linked. When we say kinase receptors, they are enzyme linked only. So that way you can remember 4 types of receptors. G protein coupled receptors. Number 2, ligand gated ion channels. Number 3, enzyme linked receptors. And number 4, cytoplasmic receptors. 4 types of receptors. We'll talk about G protein coupled receptors today. G protein coupled receptors, they have been studied in detail. This has a typical structure. We call it 7 transmembrane helical domain. 7 transmembrane. Very easy to understand. 1, 2, 3, 4, 5, 6, 7. 7 transmembrane. Outside amino terminal, inside the cell carboxyl terminal. It's a polypeptide chain. Why do we call it G protein coupled receptor? Because the effector is the G protein. G protein is a heterotrimer. Hetero means all subunits are different. Trimer means there are 3 subunits. And these 3 subunits are alpha, beta, and gamma. These 3 subunits are there. This alpha subunit is attached to ADP normally. When this ADP is attached, this remains bound to the transmembrane helix. This transmembrane structure, when it combines into quaternary structure, then it forms a binding domain on the outer surface. So here will be the ligand will bind here. Okay. Now based on this G protein, they are further classified into subunits. Actually nowadays, as I told you, we know the genetic structure, so we know minor changes also in the receptors, but pharmacologically based on alpha subunit, they are further divided into 4 families. I am saying 4. GO also is written somewhere, but some we classify GI, GO under 1. So those 4 families are GS, GI, GQ and G12. As I told you, GI oblique GO also will be written, and G12 oblique G13 also you will find. So you can remember 4 families of based on G protein, and these families have further subfamilies 2 to 10. So that way there are various types of G protein. Actually we can roughly say that all cells in our body, they have G protein coupled receptors, and more than one. Roughly you can say every cell has around 2 types at least of G protein coupled receptors. They are in abundance. So what happens? How the transduction mechanism takes place? When the ligand binds to the binding domain, suddenly what happens? The affinity of this alpha subunit to this ADP decreases and affinity to ATP increases. So what will happen? This ATP will come and replace this ADP. But immediately on replacement, this detaches from the transmembrane helix. And not only that, this G protein detaches. The G protein also detaches from each other. So what happens? There is alpha subunit that is bound to ATP and beta gamma subunit. They remain together. This now becomes the effector. Mostly this is the effector, alpha subunit. Alpha subunit bound to ATP will cause phosphorylation of various enzymes in our body, and those enzymes will cause effect, will produce the desired whatever is the effect. In some cases beta gamma also produces, we'll talk about that also. Then what happens after some time? This alpha subunit also has ATPase that causes hydrolysis of this ATP, and this will be hydrolyzed into ADP. Once this becomes ADP, everything will revert. So this is what keeps happening, zup 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 inside the membrane. This is what keeps happening, detaching, attaching, detaching, attaching. This is how it happens. So this is the transduction mechanism. But not only this, what happens to this, what this alpha subunit does? I told you 4 types, so all of them, they have different mechanisms for producing the effect. And remember, you have to remember where these types of receptors of transduction mechanisms are found, like where do you get GS, where do you get GI, where do you get GQ, where do you get G12. So we'll talk about that also. This is GS I have talked about. What happens to GS? GS and GI, both of them, they act on adenylyl cyclase. Adenylyl cyclase is situated in the plasma membrane. Always remember, it is not found in the cytoplasm. Adenylyl cyclase is floating in the plasma membrane, but the cytoplasmic site is exposed to cytoplasm. So half of it is submerged in the plasma membrane and half of it is in the cytoplasm and keeps floating. So what happens when this, say, let us talk of GS first of all. This GS binds to this adenylyl cyclase and activates it. When adenylyl cyclase is activated, this causes conversion of this ATP into CAMP. CAMP. Cyclic adenosine monophosphate. Okay. Now this CAMP, we call it a second messenger. First messenger is the, what is the first messenger? First messenger is always the drug. The ligand is the first messenger. So this is the second messenger, CAMP. This CAMP then causes activation of this pKa, protein kinase. It's a general term. There are so many types of proteins. This is so many types of proteins which will be phosphorylated by it, but this is protein kinase A. A for AMP. Another protein kinase is C, that is related to PLC. We'll talk about that. So remember, pKa means that is connected to AMP. And this pKa then causes phosphorylation of many enzymes and they will function. Like it will increase the levels of calcium in the cell. Actually, it also acts on the L type of calcium channels present in the plasma membrane, so causes entry of calcium. But mostly this causes release of calcium from the endoplasmic reticulum, sarcoplasmic reticulum. This will cause lipolysis. This will cause glycogen conversion into glucose. This would cause inhibition of glycogen synthesis. All these functions are doing what? Because GS is coupled, one of the example is beta adrenergic receptors. And what is the function of beta adrenergic receptor? Is to stimulate the heart. If you want to stimulate the activity, you need glucose. So in the liver, this GS coupled, G protein coupled receptor, they cause increased synthesis of glucose. Hyperglycemia is caused. So the effect of hyperglycemia because of adrenaline is because of this pKa in the liver. In the heart, this is causing release of calcium. And this way, these are just one of the things I have tried to explain. This is how, depending on the type of cell in which the activity is taking place, this would phosphorylate the enzymes and effects will take place. But we have to remember there are certain functions which are directly performed by CAMP also. So these are the functions which are performed through pKa. These are also functions which are performed directly by CAMP, like cyclic nucleotide gated channels are there. CNG we call them. This opens them. I told you about some calcium receptors. So we call them cyclic nucleotide gated channels which are directly opened by CAMP. Another CAMP response element binding protein, CRAB. They are stimulated by CAMP, and what do? They cause transcription, stimulation. Remember, in case of steroids, we also talk responsive element. So they are cyclic AMP response element binding proteins. Then there are exchange protein activated by CAMP, EPAC. This also has functions. So all these are functions which are done directly by the CAMP. This also you will have to remember. So amplification of signal. There was one ligand and now you see so many actions are taking place in different cells. This is amplification. And this GI, just opposite to that. Whatever is being done by GS, opposite to that will be done by GI. So need not study it separately. And remember, in case of GS, I told you there is found beta receptors. Histamine 2 receptors also has. GI, you have to find out some method to remember, but remember DAM and 2-ol. Dopamine 2 receptors, alpha 2 receptors, M2 receptors, they all have GI mediated activity. Okay. Then we come to GQ mediated activity. What is the transduction mechanism of GQ? GQ causes stimulation of phospholipase C. This phospholipase C causes conversion of phosphatidyl inositol 4,5 biphosphate into IP3 inositol 1,4,5 triphosphate, IP3 and diacylglycerol, DAG, diacylglycerol. IP3 is water soluble, so this will come to cytoplasm, and we know then gradually what will happen. In case of CAMP, I didn't tell you how the termination takes place. CAMP is metabolized into 5-AMP, adenosine monophosphate, and that is how the effect is terminated. So one of the termination was at the level of G protein, ATPase, and here CAMP also with the help of phosphodiesterase. Phosphodiesterase causes degradation of CAMP into 5-AMP. And remember, there are various types of phosphodiesterases. 2, 3 are responsible for degradation of CAMP, and phosphodiesterase 5 is responsible for degradation of CGMP. Now let us talk of CGMP also. I talked about adenylyl cyclase. There is something known as guanylyl cyclase also. Guanylyl cyclase will cause synthesis of CGMP, guanosine monophosphate. That will have, they are present, and there are at least 2 types of this CGMP are there. One is present in the membrane, but what is more important for us is that is present in the cytoplasm. But remember, don't get confused that adenylyl cyclase is GPCR attached to GS. Guanylyl cyclase is not. In the last session also I was talking about mechanism of action of nitric oxide that causes synthesis of CGMP and causes vasodilatation. This was not a receptor. So guanylyl cyclase activity is not through these receptors. That is a non-receptor mechanism. So guanylyl cyclase is stimulated by nitric oxide, not by G protein coupled receptor. You'll remember that. Guanylyl cyclase has same transduction mechanism. After that, the effector is same, but it is being stimulated by nitric oxide and not by GPCRs. So remember that. Now this GQ. So this is PKC. I told you this DAG, this causes synthesis of protein kinase C. C stands for PLC, phospholipase C. In the case of GS, it was PKA. So remember, this PKC will also have various functions like PKA, causes mitosis of cells, synthesis of plasma, migration of cells. It will have so many metabolic functions also. So this will have all these effects. And this IP3, remember, IP3 release of calcium, binding of calcium to calmodulin. So this stimulatory effect is through IP3. Ultimately what happens, this IP3 gets converted to inositol and then back to PIP2. This is how the effect is terminated. But this IP3 causes release of calcium from the sarcoplasmic reticulum, and remember, this is important. GQ mechanisms are important for alpha 1 receptors. GQ. I can give you example, muscarinic M1 receptors, M3 receptors, okay, and H1 receptors also, histamine receptors. Remember, there are one type of receptors that uses all mechanisms, that is serotonin receptor, 5-HT. 5-HT1 receptor uses GI, 5-HT2 uses GQ, and 5-HT4-7 uses GS. So serotonin receptors, they use all types of transduction mechanisms. See the isoforms of serotonin receptors. So this is how this GQ will function. Another transduction, I have told you 3. One is through GS, adenylyl cyclase. Another is GI, adenylyl cyclase. Third, I told you about GQ, that is IP3 DAG mechanism we call it. And this is the fourth one, GPCR ion channels. Now it is becoming more and more important. GPCR ion channels are ion channels which are connected to GPCR, not the ligand binded or voltage gated. They are, and very important one is inwardly rectifying potassium channels and calcium. One more thing is observed, that these ion channels are operated by that beta gamma subunit and mostly GI. So what happens in case of inwardly rectifying potassium channels? When they are opened, potassium will come out of the cell and there will be hyperpolarization of the membrane, and so they are inhibitory in nature. So it is actually seen that in almost all those places, in dopaminergic also, in adrenergic also, alpha 2 also, D2 also, and in M2 also. Beta gamma subunit is doing this. In case of adrenergic, alpha 2, this is inhibiting calcium. So these potassium channels are opened and calcium channels are closed. So effect is inhibitory of beta gamma subunit. So this is the observation, but now you will find more and more mechanism of beta gamma subunit will also come into place. But GPCR coupled ion channels, inwardly rectifying potassium channels, are found at many places, and they are opened by beta gamma subunit of GI. There are some more mechanisms. This you may not find in the books, but they are becoming more and more important. One is this G12. Sometimes we write G1213. This is a unique. This acts through RHO. RHO is a RAS family compound. So forget this RHOG. This is how actually it stimulates. Just remember, RHO is the second messenger. Like in case was CAMP. Here RHO is the second messenger, and like in those cases it was PKA and PKC. Here it is ROC, RHO associated protein kinase. This ROC causes gene regulation, and at least one receptor it has been observed it acts on. They are angiotensin receptors. Actually this has been studied a lot in cases of cardiac failure and such, and then more and more things came into picture. So this RHO is the second messenger, ROC is the protein kinase that acts on the genes, and in this case, in this receptor it has been found. But there are various types of mechanisms in which it is involved, like inhibition of ROC has been found to be beneficial in pulmonary hypertension, in case of bronchoconstriction, erectile dysfunction, and more importantly glaucoma. Actually this netarsudil is the drug that is inhibitor of this protein kinase ROC, and this is found to be effective in glaucoma, wide angle glaucoma. So this is also becoming more and more important. RHO, it is found in retina also. So this mechanism also you can remember if somebody asks you. And one more thing I will tell you, arrestin. Arrestin you will find in the books, it is written as that this causes internalization of GPCRs. That is what is written, that when it is stimulated, then it develops affinity for arrestin that is found in cytoplasm, it comes and binds to the. But now it has been found that this arrestin, it is doing it. Internalization of GPCR takes place, and then a new receptor is synthesized. So that is the function of arrestin that we know, beta arrestins. But now we know that it is also acting as a second messenger, arrestin. And it has been found that this causes stimulation of various proteins like this MAP kinase, MAPK, mitogen activated. This also has been actually studied in cardiac failure cases. This also acts on tyrosine kinase. This also RAS family of GTP binding protein like Rho. So arrestin also can stimulate Rho, and that is why these two have been found they are associated together. Nuclear factor kappa B, that is also stimulated, and because they have been found to be involved in mortality and morbidity in cardiac failures, and so a lot of research is going on in this direction, and these two are being found to be related to each other. This RAS family, arrestin and RHO. So this is also, arrestin is also one of the transduction mechanisms. So this also you can remember, at least these 6 types of transduction mechanisms you can remember. So thank you very much. Next time we will finish pharmacodynamics.